Oh, I see, I see. Awesome. So hi, everyone. Welcome to the 50th session of the MedAI Group Exchange Sessions. And this week we have Ruishan Liu from Stanford here with us to present her research on AI for clinical trials and precision medicine. Ruishan is currently a postdoc here at Stanford in the Biomedical Data Science Department and works with James Zhao. She received her PhD from Electrical Engineering Department, also here at Stanford, just this year. So congrats, Ruishan. She's broadly interested in the intersection of machine learning and applications in human diseases, health, and genomics. Her work on Trial Pathfinder, which she'll be talking about today, was also selected as the 2021 top 10 clinical research achievements and a finalist for Global Pharma Award in 2021. So thanks so much again for joining us today. And before we start, do you have any preferences on how you would like to take questions? Is it okay for us to interrupt you, or do you want it at the end?
I think either way is fine. People can just interrupt me.
Okay, sounds great. So let's try to make this session as interactive as possible. And without further ado, let me hand it over to Ruishan.
Okay, great. Thanks for the introduction, and thank you so much for inviting me. Yeah, I'm very excited to be in this session. I've also seen a lot of videos from MedAI in YouTube, so this is also my first time to be here online. So yeah, hello everyone, I'm Ruishan. So today I'm going to talk about our projects for AI for clinical trials and precision medicine. Yeah, as just introduced, this is the work with my advisor, James Zhao. Okay, let's get started. So if we think about AI for medicine, we can think about AI using for the whole medicine. So here we say medicine is the drugs medicine. So they usually have 3 steps. So for the drugs, you first develop the drug, and then the drug companies develop some clinical trials. And once the drug is on market, we'll further use that drug in the population. So particularly, so for the drug development parts, AI has achieved a great success. So because like we have a lot of well-defined tasks in that step for the drug development. For example, for drug screening, we can use AI to predict bioactivity or toxicity. So those are well-defined, supervised tasks. And also for molecular design, I think like most people may heard about the DeepMind AlphaFold, which is an exciting breakthrough to predict the protein structures. So basically, if you think about the AI for medicine, this pipeline, we will achieve a lot in the very beginning, like in the source, when we do try to develop the drug. But for the last 2 parts, especially how AI can help clinical trials design, or how AI can help to give the patients the drug, are still like very early stage for AI. So this are also the main topic we want to talk about today in this talk. So our meaning talk about our 2 projects. So first is we use AI to optimize the clinical trial design to help to get more inclusive clinical trials. And the second one is we try to do a systematic pan-cancer analysis to give a guide for the precision oncology. Okay, so I'll talk about those 2 projects in parallel. So maybe I'll go to the first one, so AI for clinical trials. Okay, so before I go into the particular project we are working on, maybe I'll also give you some basic knowledge of what clinical trials are and also how we can contribute to this. So the very first is clinical trials. What a typical clinical trials look is really you will recruit patients and you randomly assign them in 2 groups. And some basic knowledge is the median cost of a phase 3 trial is around $20 million. So it's really very costly. And also the average time for enrollment is 8 months. And 86% of clinical trials fail to complete recruitments within targeted time. So basically, the trial recruitment process and the trial itself is very slow. And also, for each month delay, it costs more than $600K. So basically, it's slow and it makes the trials to be even more expensive. So that leads to a very large problem for clinical trials, which is the trial enrollment is very slow. And you may wonder, yeah, so we know the facts, it's slow and make it to be even more expensive. So, but what leads to that facts? So actually, like one key factor that leads to the slow enrollment is the strict eligibility criteria. So basically, not all the patients who apply for the clinical trial could be enrolled. So they usually have very strict enrollment criteria. And for example, this is some statistics. Basically, for some non-small cell lung cancer trials, the statistics show only about 20% patients they are eligible for analyst trials. So to that step, you can see those strict criteria would exclude a lot of patients, which leads to slow enrollment and costly trial. And towards that, the NCI clinical trials program concluded that the eligibility criteria for all cancer clinical trials should be simplified in order to require minimal input at the time of registration of individuals. So you can see there's a strong need and a consensus to simplify the criteria, but it's very hard. So maybe before I go into tell you how we can relax them, we can see what at this stage, at current stage, how those strict eligibility criteria are decided. So here is one example. So this is from our collaborator, Roche. They developed some new drugs, and this is their working protocol drafts. Basically, they have some working drafts like a shared Google Doc, and we are very lucky to be in this collaboration, so we can see their working drafts. So this is just a screenshot of part of the inclusion criteria they use for this particular drug. And basically, what people do, the clinicians do to decide the clinical trial eligibility rules are they just first simply duplicated from previous trials as a start, just the black text you see here. And then they just modified based on their own experience. So for example, so this is the real drafts we've seen. So they have some rules for ANC, the lab value, and someone would modify it just as what we usually do in our collaboration. Like we have a shared Google Doc and people will make comments on it. So the first clinician will say, oh, can we change this threshold to be 1,000 to relax it? A clinician B will say, oh, probably there's some risk. And clinician C will say, oh, I agree to relax that. So basically, if you look at, this is a real example. So how those drug companies decide those rules are pretty at their own experience. So pretty anecdotal. And this is to decide which threshold. And they may even say, also need to design which rule to use. So in this case, is they copy the rule from some previous protocols, and they just delete the rule as a whole. So this is gives you just in real world what are those trials like actually end up with the rules. So basically here each row is one trial. So here we have 10 clinical trials for non-small cell lung cancer, and each column is one rule. So there are different rules. There are rules for biomarkers, rules for diagnosis and laboratory tests, etc. So here, the yellow block means that trial has that rule. So basically, you can see for some rules, like they are consistently used across different trials, but there are also a lot of heterogeneity in the rules. What are the rules used by the people? So the FDA concluded that current eligibility criteria, sometimes excluding certain populations from trials without strong clinical or scientific justification. And also the unnecessary exclusion can lead to fail of the generalizability of the drug's efficacy to the real world population. So you can see there's actually a very strong consensus, and also the current stage, what current people would do for the clinical trials are very anecdotal, like based on their own experience. But the difficulty is the performance on the excluded patients is always unknown. So this is actually a counterfactual problem. So basically, if you imagine some clinicians, they may decide criteria before in some trials, but they will never know what will happen if they relax those criteria. But in the future, they may just continue to use those rules. So here comes to our project. So we propose a method, also a computational framework. We call it Trial Pathfinder, where we use real-world data and AI to design the eligibility criteria. So I'll give you some high-level idea of how our framework works. So the idea is from the Trial Pathfinder, we can emulate the clinical trials from real-world data, because the difficulty for the clinical trials they are counterfactual. We can never know what people do or would end up with if they should be excluded. So here is in the real-world data, we get all sorts of patients. They can get all different of treatments. So we can emulate the clinical trials from the real-world data. Basically, we can generate synthetic cohorts from the real-world data. And then the next step should be more familiar with machine learning community. So after you get those synthetic cohorts, you can just even like say evaluate individual rule or optimize to some optimization for the clinical trial. So the very first, we want to emulate clinical trials. So before we see how we emulate clinical trials, we need to first give like short background of what are the clinical trials. So basically, the clinical trials, generally we have two groups of patients. So the treatment arm and control arm. And those patients, they will be randomly assigned into one of those two arms. So for example, in this case, the treated arm is drug A, which is a new drug the drug company want to test on. And the control arm is drug B, usually the standard of care. And so in the real world data, we will have patients with all different of demographics. Here we use sex just as like a representative feature, and they will take all different drugs. And so what we can do is we can generate synthetic patient cohorts from this. So basically, the treated arm is all the patients who took drug A, and the control arm is all the patients who took drug B. And we can also emulate the eligibility criteria. Basically, like we can emulate, we are doing the clinical trials, we just exclude the patients in the gray who do not meet the eligibility criteria. So basically, to that step, you can see we can generate synthetic patient cohorts for treatment arm and control arm. And next step is we want to compare those two groups. Basically, we want to compare whether drug A is better than drug B. So what we usually do for clinical trials is for the survival analysis. And here, what I show here is the survival curve. Basically, the x-axis is the time to the patients get enrolled, and the y-axis is the overall survival. Basically, at time 0 is when all the patients just get enrolled, all the patients are alive, so the overall survival is 1. As time goes by, there are patients dying, so the curve decreases. So here you can see in the treated group, the patients dying just slower than the control group. That means actually like the new drug is expected, it performs better than the standard of care. And also, because what we use is real world data. So we also do some extra statistical adjustments for us. So basically, we want to emulate the blind assignments to the 2 groups. So we use technique called inverse probability of treatment weighting. So the idea is so the idea is to say, for each patient, we give it a weight to see how likely it would appear in the other group. Basically, this is also a standard technique to emulate the blind assignments. And also, we use a general metric hazard ratio to measure like how well the drug is. So this is what we do to that step is or following what the normal clinical trials would do. So the hazard ratio basically just to capture the hazards for the drug compared to the control drug. So basically, hazard ratio less than 1 means the treated arm performs better. If hazard ratio larger than 1 means treated arm performs worse. So in this case, the treated arm performs better and the hazard ratio is less than 1.
May I ask a
Okay.
question here?
Yes.
So when is IPTW used? Do you use it to weigh the Cox model during survival analysis or? Yes. Okay. Thanks.
Yes, exactly. We use that to weigh down the Cox model. So basically, when you feed the Cox model, you just give the data points different weights.
Okay, thank you.
Yes. Yeah, so basically to that step, you can see we know how we can do to generate synthetic cohorts from real world data, and we can emulate the clinical trials, like just run the clinical trials in silico and to see how the drug would perform on those population. And the next step is like the main task we want to do is we want to evaluate the clinical trials, like their own eligibility criteria, and make some recommendation. So basically, here we can change the eligibility criteria in silico. So for example, here we can see if our input is 2 rules, like rule 1 and rule 2, and how many patients would left. For example, here is 1,000 patients, and within those 1,000 patients, we can see the drug is efficacy, the hazard ratio is 0.7. And also we can do is we can delete rule 1, we can only look at rule 2. And then like we can see there are more patients getting included, but the hazard ratio increases. So that basically saying like, even though rule 1 filled out a lot of patients, but it's actually good for the signal of the drug. And we want to quantify the contribution of each rule more rigorously. Like this here in this example, you see rule 1 is good, but here we want to quantify it more in like a more rigorous mathematical way. So we use Shapley value. So Shapley value, the idea of Shapley value is to say it within a set, we retrieve the contribution of one element by averaging like or sorts of different combinations of the rest of rules. So Shapley value is actually now is widely used in machine learning community, for example, to evaluate data points. So here in our example, in our case, the data points is rule. So here we use that to evaluate each rule of the eligibility criteria. And here, the value less than 0 means that rule is good to include. Basically, if you include that rule, that leads to hazard ratio to decrease. So yeah, so if you're not familiar with those terms, hazard ratio, Shapley, you just keep in mind, like if it's less than 0, it means it's good. Yeah, so to this point, you can see you already have a basic knowledge of what our pipeline is. So we emulate the trials and we change those rules in silico and we evaluate it with Shapley value. And here we implement a Flatiron database. So this is a collaboration work with Roche Genotech. And here we analyze 61,000 patients with non-small cell lung cancer. And also we use 10 clinical trials as an example. So here is the main results. And also this is what those clinicians are excited about. So basically this is Shapley value analysis. So here each row is one clinical trial and each column is rule, like one rule. And here the color indicates the Shapley value. So if it's red, it means larger than 0, means if you include that rule for that trial, that leads to an increase of hazard ratio, which is bad, and the blue is good. So so basically, so this I'm not sure like whether like our participants like backgrounds are. So if you are like me, we are more like computational side. So for us, this is just a raw results, but for clinicians, this actually would give them a lot of information, especially when they want to design clinical trials in the future. They may just look at the trials which are similar or drugs in similar usage and look at the contribution of different rules and use that as evidence for their future decision. Yeah, and actually what we found is a lot of rules in the rights. So they actually do not have a like strong influence on the signal. So basically it's okay to include or not include. So our recommendation would be maybe just not include them because like they would exclude patients, but they do not really contribute to the drug signal.
Can I ask a quick question actually before you dive into the results? This is super cool, but I guess I'm still missing how exactly the synthetic data is obtained. I think you already explained this before, but maybe you don't mind just going over that a little bit.
Oh, yes, my bad. And so our data is, so basically this is a very rich database. Basically we have the EHR. So basically you can extract the treatments and the patient's survival information, patient's visits, and also patient's demographics, lab values. So basically everything you may want, like they are in the database. And particularly for this projects, we'll need to extract their treatments, survival, and also for their different rules, we'll need to pretend they are entering the clinical trial. So we'll look at their different, for example, lab test results near the time when we want to do the analysis. Yeah.
I see. And so in this database, you already have information about whether the patients took a certain drug or not. It is, and then based on that, you if they have taken a certain drug, then you can assume that they are in the like they're not the control, but if they've not, then it's the control. Is that the gist?
Yes, exactly. Yes.
So can I ask, just to clarify that point further, so this data is for drugs which are already in the market. That's why you have patients who have taken these drugs. So okay. So it's kind of, you're getting the data from the future.
Yes, exactly. So yeah, that's a very good question, because we will never know what will happen for the drugs not on market or like roughly new drug. So here what we do is we analyze, basically we can analyze all the previous clinical trials to see how well their eligibility criteria are. You see what I mean? Because for all the drugs,
so it's like, so it's like saying, okay, when these drugs were being tested, these conditions could have been relaxed. They don't really seem to make a difference in this, future trial. Okay, yeah, that makes sense. Thank you.
Yes. That's a very good point. Yeah, so basically you can just run this very systematically for all the old drugs and all the old clinical trials, and you can get some knowledge like what we get here, like a lot of rules, they're just consistently not contributing. And yeah, and also add to that point, for the application, it also say, because a lot of trials, they are not really looking at new drug, but they are the drug look at different usage. They will like say, for example, look at different histology stuff. So basically for those old drugs, you can directly use those old trials for that drug as reference. For roughly like new, like a total completely new drug, basically what would recommend is to look at all the drugs which have similar for example pathways or like mechanisms. So those are also good reference. Okay, great. Yeah, so apart from that, we can also do some simple thing. Basically we just for those old trials, we just use look at all the criteria which have good Shapley value, and here is what we end up with basically. So for the data-driven criteria, we can double the eligible patients, while we can even reduce the hazard. So what this actually means is, so for those trials, if you use it in a more optimized way or more relaxed way, you can double the patients, while do not really harm and even could reduce the hazards. So basically, it means if you do this, that would accelerate your enrollments, right? Like the twice of the speed. And also the drug will get tested in a broader population, because you will have like more generalizability while in some markets. And also, if you just recall, like a very hot point for those protocol design is to design different thresholds, because people do not really know, whether it's 1,000, 1,500, they do not really have evidence for that. So we can even also do this. We can relaxing the threshold in silico. So here is one example. So this is threshold for bilirubin. For those 10 trials, they have like 2 different thresholds. And what we can do is we can change the bilirubin threshold in silico and to see how that Shapley value changes. So what we'll see is, like a quick conclusion is, we found it's pretty safe to just relax your thresholds. And also we will make this to be an internal tool within Roche Genotech, like within the company, because this data set is inclusive for them. And also like their drug company, they are using this to design the drug. So we just make this to be an internal tool for them for their drug design. And yeah, I think it's very it's also like great for us to get their timely feedback from their team, because they are really design the drug, design the trials. And also, so this project is very honored to be selected as finalist for Global Pharma Award 2021, and also the top 10 clinical research achievement last year. Yeah, so basically, to this step, I'll just conclude my first projects, like first half of the presentation, where we use AI to help the clinical trials design. You can see we can use AI to emulate clinical trials, and also to help make the trials more inclusive. And the next part, I'll move on to use the large real-world data to get more knowledge for precision medicine. Okay, so
can I ask a quick question before you move to the second part?
Yes.
I thought the first part was super cool. The one question I had was, did you do any analysis on the demographics of the patients that were now included because of this relaxed criteria versus like what they initially had, and what there's some interesting findings about?
Yeah, thanks for mentioning that. That's actually a very good point of our work, because what we find is we actually, if you relax it, you will get a more diverse group. So basically what we found is those clinical trials usually end up more restricted. If you relax them, we'll typically include more patients, like the Black patients, and also we'll tend to include more female and also older patients. So basically, so that's also like a one point, because I didn't put it on, but basically the strict rules would filter out minority groups, like more likely to filter out minority groups. So that doesn't really mean like they really make the rules to exclude the minority groups, but it's because say for example some lab values, it's just more strict for female than male, but they just use the same lab rules. And also for different ethnicities, for different races, they have different demographics, but it's just like the old rules usually filter out a lot of minority groups. Yeah, so that's cool. Actually, yes, yeah, so that's actually a very good, also another good motivation for why we really want to relax it, because currently it's not very a good representative like for the whole population.
One question, basically, I mean, do these drug companies and people designing these clinical trials, do they have any, incentive for relaxing this criteria? I mean, if you put so many restrictions on your trial population, does that mean when your drug hits the market, it will be used on a more restricted population, which kind of match the clinical trial population criteria? I mean, so if you relax this, you can potentially be targeting a larger population. Is that so?
Yeah, so I think that there are like two parts of it. So the first part is, so I think that is a little bit tricky. So even though, because we know like all the drugs, they will have strict criteria, but when they are on market, that's actually, those would give to all the patients. So basically they are not really restricted to those labels. So for example, for the
okay, so I mean, actual population where it's used, it doesn't have to match the clinical trial population necessarily.
Yeah, exactly. Because for a lot of rules, for example, if they really shared across. Let me find across all the trials. So basically, for example, like patients older than 60 years old, they will never enter those clinical trials, but in the real world, there are always patients who are older than 60 years old, so they will get the drugs anyway. So that's also why we will have signal, because in real world, we will like the doctors will give the drugs to patients outside the labels of the clinical trials.
Okay.
Yeah. So actually, like the incentive or the motivation for the drug companies to relax is mainly for the costs, because the clinical trials itself is very costly, like just in the very beginning, like because it's like $20 million in average, and also the delay. So basically, the cost of it's like a lot of the proportion is from the enrollment process. They will need to like a very long process for the enrollments, but like say, because in general, like if you filter out 80% of patients, you will need to wait for much longer. And also we have the statistics like near 90% of clinical trials, they fail to complete the recruitment. Like they fail to recruit enough patients. So yeah, so basically for the drug companies, their main consideration is for the cost, because if you can have relaxed criteria, that could hugely reduce your costs. And for our scientific parts, we actually also want them to relax it, because that would have a like signal for more general population. So because when you are markets, that actually gives a more generous ability.
That makes sense. Thank you for explaining that.
Yeah. All right. So then I'll move on to our second part. So use the real world data, large real world data to do some precision medicine analysis. So I'll give some like also like basic explanation of what is precision medicine. So the definition is, so before what we usually do is we do one drug fits all. So for example, if you have this type of cancer, this stage, I just recommend you to a certain of drugs. But as we have more data, as this big data area, we are capable of to do more individualized recommendation. So more strict definition is from their precision medicine initiative is so this is approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle for each person. So the environment and lifestyle would be a bit hard to capture and also be slowed like in the research. So in our project, we'll mainly focus on the genes. So basically how to personalize your treatment based on your genomics profile. Okay, so here is also some statistics. Basically, a survey in 2018 from the US shows that only 8% of patients, they are eligible for genome-targeted therapies, and only 15% of patients, they are eligible for genome-informed therapies. So basically, this is to say, now we have like very limited knowledge about like, say, given the patient's genomics profile, what are the good therapies you could give. So basically, this is a very simple but very important question to ask. So how patients with specific genetic mutations respond to therapies. And here I just describe our model, like a simplified version. Basically, we want to measure the interaction between the gene mutation and treatments. And here is the hazard function, basically the hazards of dying at each time step. And here is you can see like basically this is a also the Cox model, and you have a mutation term, treatment term, and what we care about is the interaction term. So what the interaction between the gene mutation and the treatments, and also adjusted by a set of confounders like the patient's demographics and health conditions, etc. So basically what we care about is, we call this term as mutation treatment interaction hazard ratio. So if it's less than 1, it means the treatment effect is better in patients with a gene mutation. So if you have the patients have that gene mutation, they are more recommended for that treatment. So still, we implement on the Flatiron database, where we have 8 cancer types with at least 1,000 patients, and we just examined 605 genes. So here is the the statistics. So the figure A is, so each row is 1 cancer type with a number of patients, and each column is 1 gene. So here is just like a featured genes from these 600 genes. And so the color of it is the proportion of positive patients within that cancer type. And also like the small digits within them is the number of patients who are positive. And figure B is, because for the mutations, you will have different types of mutations, type point, rearrangements. So figure B is what the proportion of each mutation for those genes. So here it just gives you some basic sense of what the data look like. Yeah, I will give you some like example case study to interpret the mutation treatment interaction, or why we care about it and how we want to use it. So basically, one example is we want to look at, for bladder cancer, how gene APC interacts with immunotherapy. So basically, in the real world data, we can look at all the patients who are on immunotherapy. So basically, we look at the group of patients, they took the same drug, they all took immunotherapy, and we can compare the performance of patients with APC mutation and without the APC mutation. So here you can see the orange curve, patients with APC mutation, their survival curve is above. Basically, as time goes by, their like death is they're dying slower than the other group. So basically, for the hazard ratio it means like the hazard ratio is interaction hazard ratio is less than 1. And if we look at in another perspective, so if we look at all the patients with APC mutations, so basically this is to say, for example, you are a patient, you already have APC mutation, you want to compare immunotherapy versus other therapies, right? And this is also to say the orange curve is still above the blue curve. This is also to say like the interaction term should be less than 1. So basically, this is what it means, what the mutation treatment interaction means. And so basically what we compute within our model, the hazard ratio is also less than 1 with the 95% confidence interval. And also we have some validation on clinical trials. Basically, because to this point, we are looking at real world data, there could be a lot of confounders, and also it's not random mill site. So we can validate it on clinical trials. So basically there are a bunch of clinical trials. They happen to have some genomics profile, but this is pretty rare in reality. But particularly in this case, we have that and also get enough patients, and the hazard ratio is less than 1. So basically, it's just a validation for the results. And so what we do is we repeat this process for all the mutations across basically across 600 mutations, 600 genes, and all different treatments, and across all different cancer types. And what we do, we can reports with like all the hypotheses it generates. Like say so in this plot we just give a subset of the mutations that are significantly predict the outcome patients on specific treatments. Basically the hazard ratio p-value less than 0.05, and we have some FDR control. So here basically each block is one cancer type, the non-small cell lung cancer, and also different small blocks are for different treatments, for example, immunotherapy, pembrolizumab. And here, the gene name, like the size of gene name, is proportional to the number of patients who are positive. That means if the gene name is very large in the graph, there are a lot of patients in the real world who harbor that gene mutation. And the color corresponds to the interaction hazard ratio. So if it's red, means that's a good interaction, a bad one, so hazard ratio larger than 1. If it's blue, it means it's a good interaction. Basically, if you have that mutation, you are more recommended for that treatment. So we can review this as a hypothesis generation. We have this, we have like a full list, but this is a subset. It's for clinicians to look at, or say, so we would say like a lot of those already have some clinical evidence before. Now we also add in more hypotheses, like more rarely mutated genes, and also like new treatments. So to actually put this as a public tool online, so we put this as a public website where we put all our results in, like for the all pan cancer analysis, and and also we have some other features. For example, what I just described is mutation-treatment interaction. We also have some features for mutation-mutation interactions. That is to say, the co-occurrence of the mutation, how would that lead to the performance?
Oh,
Yeah.
I actually had a question exactly about the co-occurrence part.
Yes.
So in your analysis, like if you find more than one mutation, and so how do you actually analyze it? Like how do you marginalize what contribution is from one which mutation?
Yes, yeah. So that's a good question. So the results we show here is like, say we look at we do not think about the causal so the results, so this version of results, we do not think about the causal. So it's like whether that is caused by the specific gene mutation or that one. That is to say, like, say for example, if two genes, like in the most extreme case, if gene A and gene B, they always co-occurs, right? And basically, like gene A, just if you have gene A, you must have gene B, and vice versa. But we know like the real causal effect is from gene A. But so in this results, we do not really consider of the causal. So it's like even if it's not caused by gene B, but the observational study shows that if you have gene mutation from gene B, you're more likely to benefit from some treatments. We'll just report that.
I see.
Yeah.
And so you might have, so let's say you have a particular mutation A, and then so let's say a big portion of the population with mutation A actually benefits from, let's say, a treatment, but then that population also contains mutation B. So it's hard to say whether if somebody has only mutation B, whether it's from, like, whether they will benefit or not. Is there yes. some ambiguity there, or is it usually, like, was it mostly clear, like you'll have the percentage of population with mutation B and without mutation B? Was that always clear?
That's roughly basically because what you will get is a very comprehensive list of what different types of genes their contribution are. So if you have both A and B, that might gives you a very strong signal, right? Yes. So yeah, we actually that's a very good point. So we're actually considering, we consider different types. So we also have a version of adjusting for different other mutations. So basically we can adjust it for, we have a version of results for that. But we get already like advice from our clinicians because they found that would be a bit vague for them to interpret. They found this would be more straightforward because it's pretty easy. Like they just look, oh, like this highlighted ones, if they found this would be better. But we do have a version of adjusted one. So we actually also have but this is ongoing projects, maybe like a more machine learning or AI studies to have a predictive model. We actually could say predicts the performance of different treatment based on the whole gene profiles. So basically you can input your demographics and profile. I can tell you like what's for different treatments, what your performance would be. So that would be like very, so that would include all the effects. But for this project is for the clinicians, because that is also like a very common situation for machine learning community, because we can run very complex model, have very good prediction considering all the things, but they do not really trust or feel very comfortable with it. But if we so the project just ends up in this way. Basically it's like a trade-off also with the clinicians. So because for example in this one they can just very straightforward, it's very straightforward for them, like, oh yeah, this gene, if you have like if the patient have that gene, you're more likely to benefit for this. Yeah, so that's a very good point. Yeah, that's also like how our projects evolve. Yeah, so but I feel like it's very great to get suggestions from clinicians, because they are the people who really want to use or will use this. Yeah, but
Makes
that's
sense.
a very good considerations.
Thank you.
May I know how you determine the confounders? I think it's previous one slide before this. In your in your interaction term, or like in a
I see.
sorry.
Yeah. So basically we include all the things we are able to include, basically like the patient's demographics and age, gender, race, and also like the year of the treatments, and also we will test it on lab values, those kind of things. So basically so how do we determine it is from two sides. So one side is what are the available on the data, and also we run to see if there's a significant difference. And the second one is we get it also reviewed by our collaborator clinicians, and also they will sometimes also tell us like what they think might be a good confounder, and we'll try to find that from the EHR as well. Yeah, so basically the confounders would be like also data driven and also from their evaluation.
I see. Makes sense. Thanks.
Yeah. Right, and yeah, basically we kind of want to make this project to be a more hypothesis generation. So our related clinicians, they could check, and so for example, this could help you to design further like clinical trials even and stuff. Yeah, so maybe I'll just quickly conclude. Basically our two works is fall into the large framework of AI for medicine, and we focus on the last 2 steps for this streamline. And we can use AI to help design the clinical trials, and also use the large real-world data to give like more systematic knowledge for precision medicine. Yeah, thanks for your attention.
Awesome. Thanks so much, Ruishan. I think that was a great talk. Are there any more questions from our participants today? Oh, actually, before that, maybe we can all give a round of virtual applause as well. So thanks so much.
Yeah, thanks for all those questions. I think those are also, like Michelle, the key points to the projects.
Absolutely. Yeah. I guess if there are no more questions for now, we can end the session by thanking Ruishan again. We'll have the video up on YouTube later tonight, so you can always feel free to check it out. And if you have more questions or discussions, please feel free to contact Ruishan, or you can contact us and we'll be happy to put you in touch with her. Yeah, so we'll see you all next week, and thanks again for joining today.
Great, thank you.